a few things about schizophrenia and why I'm interested in it. So I saw this fact, the first thing up there, in that 1 in 100 people in America experience schizophrenic symptoms annually, which is over 3 million. And I've always assumed it's a niche disease that's relegated to insane people, but the truth is it happens a lot, and these people can't really live normal lives. You have hallucinations, which are things that don't exist, delusions, which is thinking your hallucinations are real, and thought and movement disorders. Schizophrenic people typically have really erratic movement and can't organize their thoughts. So these people, cannot live a normal life. And okay, so probably confused about this too, walking in, kind of creepy, but this is a self-portrait of someone who has schizophrenia, and they believe this is accurate of what they look like. This shows their distorted image of reality. So that's just showing you, how schizophrenia can affect you. And treatment is basically just lessening the symptoms. So you can take antipsychotics, which will decrease the severity and amount of hallucinations. You can go to psychological rehab, which can help you integrate into society. But in the end, you have to live with it for the rest of your life. So the paper I'm presenting on is Increased Despindin 1B isoform expression in schizophrenia and its propensity and agrosome formation, or the buildup of misfolded proteins in the brain and its connection to schizophrenia. So what the researchers first did is focus on a protein called Despindin 1, or dystrobrevin binding protein 1, which they know can form these things called agrosomes, which I'll explain in a bit. But first is protein. So this protein is commonly found in the block 1 complex, which is a protein complex found in musculoskeletal cells, and its purpose is of structure. So it connects the cytoskeleton, the interior skeleton of the cell, with other cells outside of it. So what's important to us is that it's also found in axons, which is a part of the neuron in the hippocampus, which has been shown to play a part in the formation of schizophrenia. You can see it like right there, that little curvy thing. And there's 3 isoforms or types of this protein, A, B, and C, and they all basically do the same thing, but they're slightly different. Okay. So with this representation here, this is with human patients. The white bars are people who do not have schizophrenia. The black bars are people who do. As you can see here, this difference is actually, okay, so this means not significant. This difference is not significant. It looks like it is, but there's too much error, so, for all intents and purposes, that is the same. There's no statistical difference between the total amount of despindin in schizophrenic people's and normal people's brains. But there's much more despindin 1B, which is one of the 3 isoforms, in people who have schizophrenia. All good? So now to explain what agrosomes are, what I said before. So when a cell produces too much protein and some of it becomes misfolded, or it can't function basically, cells will send it to proteasomes, which are small molecules that break down proteins. But if the proteasomes are overwhelmed or not working, the cells will group the misfolded proteins into things called agrosomes, which are basically just bundles of these proteins right next to the nucleus. And this is important for schizophrenia because they've been shown to cause or help cause other mental diseases: Parkinson's, Alzheimer's, Huntington's, and Lou Gehrig's disease. So what we can see here, this is using GFP, green fluorescent protein, which is just a marker. So this is a normal distribution for despindin, what this should look like. And 1A and C have a normal distribution throughout the cell. But you can see here that despindin 1B is all clustered, almost all the protein is clustered right next to the nucleus of the cell. That's an agrosome. So that means that the cell cannot perform its breakdown of this protein, and instead it's just grouping it up all near the nucleus. And these are mice neurons here. And then this is using a different type of cell, COS1, which is a monkey, cells from a monkey. So in this case, what the researchers did, instead of introducing the protein, they introduced the DNA for the protein, and that's what a plasmid is. So they did it for the 3 different isoforms as well. And you can see here that this is A and C, that the more plasmid you add of A and C, you don't really have more agrosomes, because this axis is the amount of cells with agrosomes. But as you add more 1B, what's this right here? Like more as you go to the right, you get more and more agrosomes. So basically, the more despindin 1B is being produced in the cell, the more chance you have to form an agrosome. Okay, and then, okay, this may look a little confusing, but this is a Western blot. And the most important thing you need to see is the S and the I. So S is soluble, which is normal distribution throughout the cell. I is insoluble, which is an agrosome. And the first 2 columns, these are A, second are B, third, and the last ones are C. So what you can see is that C did not form any insoluble agrosomes, but A and B did. They all have soluble portions, but A and B also have portions that are completely insoluble. So knowing this data right there, can anyone explain what this means? Jackson.
Okay, so you just said that C doesn't have any soluble portions, but A and B do.
Yep.
And G is a graph of the percent of all that you measured, how much of it is soluble. It's basically just giving a numerical representation of the part on F. And since none of C is soluble, all of it, or none of C is insoluble, 100% of the C portion is soluble.
Yeah, exactly. It's just a numerical representation of this data right here. All right. So, but in this, so we were talking about despindin 1B before, but we can see here that despindin 1A, easier to see over here, also has insoluble portions. So the problem is they both have this domain called a coiled coil domain, both despindin 1A and B. And what can happen is this domain can interlock with itself on other proteins. So, kind of like connecting the two. We can see here, this is another, using GFP again. This is the concentration of despindin 1B in the cell. And you can see again, it's forming an agrosome. But then we also see that despindin 1A is forming an agrosome in the same exact place in the next picture. So what that means is that despindin 1B is recruiting despindin 1A to also form agrosomes near the nucleus. And with that happening, that means there's an even larger percentage of the cell's despindin that is taken out of its normal use and being put in agrosome, which is unuseful. Okay. So this first graph over here is just showing that if all of your despindin 1B is functioning normally, you don't really have a chance to form any despindin 1A agrosomes. But this is with all the despindin, almost all the despindin 1B aggregated in a cell. You see that actually the majority of despindin 1A will follow suit and aggregate as well, representing an even larger portion of the total despindin that is an agrosome formation. Okay, let's talk about the next part. We need to know what neurons are and certain parts of them. So neurons are brain cells. We know that. And we need to know what neurites are. So neurites are dendrites, which are these little things right there, and axons are this long portion. These are 2 neurons. And neurons make connections between each other when an axon comes in contact with a dendrite at a space called the synapse. And axons and dendrites together are just called neurites. So if there's no connection, no signal can be made, right? And, all right, so moving on to this question, why would a lack of soluble despindin 1 inhibit neurite growth? This is like thinking back to almost the beginning of my presentation. So, does anyone have an answer?
Okay. Well, if the despindin 1 is forming agrosomes, that means that
Yes, yeah, that's it. So I said before that it's commonly found. What?
No, it's just the quality of the answers is very high.
Well, I said before that it's commonly found in axon bundles in these neurons. So there's basically the cell needs dystrophin in its axons to grow them, to inhibit or not inhibit to what's the word? Just to grow their axons basically or their neurites. So moving on, why would the length of the axon matter in regards to schizophrenia? Anyone answer that? Yeah, I know Jackson probably can. Bingo.
Well, schizophrenia is all about hallucinations or not all about, but if it involves hallucinations and delusions, that would suggest that there's some sort of impairment of thought going on. And if axons or neurons can't function when they're not touching each other, i.e. when they don't have neurites, and if lack of soluble dystrophin means there's no neurites, then thought in one way could be conceivably impaired.
Yeah, exactly. If there's reduced neurite growth, yes.
But couldn't it just be the opposite? Like, couldn't you have crazy thoughts if you have
Too many connections?
Yeah.
I guess, but that's not what this is saying. I mean, they didn't look into that. But yeah, Jackson, you're right. So, moving on. We can see here, we can see that in action here. So these are wild type mice. They naturally have dystrophin in their brains. And this up here is a neuron that has normal dystrophin distribution. And you can see this long portion is an axon. But when a lot of dystrophin 1B is introduced and starts forming agrosomes, there's almost no neurite growth at all. You can see it's tiny compared to the one up top. So it has trouble making connections with other neurons, and that impairs neural circuitry, which could then lead to schizophrenia because there's not as many connections between cells. And then, alright, this is sad. There's a breed of mice called sandy mice that all have schizophrenia because none of them have dystrophin in their brains, so they have vastly reduced neurite growth compared to other mice. And they all have cognitive and locomotive disabilities. And the researchers use them in comparison to wild type mice to see how, basically to get numbers on neurite growth. As we saw before, a visual representation, but this is graphical. So there's two different controls for these graphs. You can't really compare them directly, but we'll start with the sandy mice, SPY over here. So they normally don't have any dystrophin in their brains, so they already have reduced neurite growth. So when dystrophin 1B agrosomes are added, you can see there's not really much of a difference between neurite length. But in wild type mice who normally have dystrophin, when lots of dystrophin 1B is introduced and it aggregates, neurite length drops dramatically. And again, that's doing the same thing, impairing neural circuitry. So, anyone tell me what the difference basically is between wild type and sandy mice, and why this would matter? I can answer my own question. That's it.
When wild type mice get introduced to bad dystrophin, so dystrophin that doesn't work, they basically have the same exact neuronal effect, like loss of neuronal circuitry as the mice without dystrophin at all. So that shows that when dystrophin is not working, you have schizophrenic.
Yeah, exactly. So not having dystrophin and having dystrophin aggregated is, they're almost the same. So basically the pathway that this paper follows is that you have lots of dystrophin being produced in the brain. It's a normal function if it functions normally, but if it doesn't, it aggregates. These agrosomes of dystrophin 1B recruit dystrophin 1A, representing a majority of the total dystrophin in your brain. and then that makes neurite growth lessen, and then cells can't connect with each other as well, and you have schizophrenia. So, yeah, because they form fewer connections with surrounding neurons. So, in the future, because I mean that's a conclusion, but doesn't really get us anywhere, I guess, if you just look at it shortly. But the next thing we need to do is start studying this in vivo, studying human cells as well, because all that we looked at is mice and monkeys, and they were almost all cultures. So we need to test this in human people, or human patients, right? And maybe use drugs that could target the aggregation of this protein. And in the far future, gene therapy might work, but don't know yet. And I think the most important thing about this is that more people need to know about the disease, because usually people just treat schizophrenia as some kind of small, weird disease that only affects crazy people. And the fact is that there's millions of people who have this problem, and I feel that with more public awareness about it, more research would be done. Yeah. So this is my thank you slide. Jackson wanted his name on it. All right, that's the end. Any questions?